Hello and welcome back to the second part of Chapter 2. You may be clicking on this right after the first part, or you may have taken a little bit of a brain break since this is a pretty dense chapter, very small to the level of ions and all of that stuff. So, either way that you handled it, welcome back. I did want to let you know that for this second half of Chapter 2, and as well as the first half of Chapter 5, there were some technical difficulties using the Zoom links. So I'm using a new program called Screencastify, which means you may be hearing this or seeing this as a YouTube video or something like that. And it seems to be a better program, so we're going to go with it. The other lectures from the first module are still the Zoom links, and maybe eventually I'll transfer them over as well. But this lecture and the first lecture of Chapter 5 ended up having no pictures. So there were some technical difficulties that I did not want to give you guys just a lecture with no slides. So let's review really quickly the movements of the action potential as the electrical signal goes down the cell from the cell body down the axon to the axon terminals. And then we will get into refractory periods and the second half of this lecture, which is the actual chemicals themselves and how they get from point A to point B, how they go from cell to cell, and what do they do. So as a reminder, what we see here, and I'll hit presentation mode. What we see here is hypopolarization, or what I call depolarization. Here we are at resting potential, which is around negative 70 millivolts. If you have a little bit of voltage come in, say 1 or 2 sodium channels open, and you get a little bit of sodium coming in, that makes the cell a little less negative. But it doesn't reach a threshold or anything like that. This is what we call the local potential, and that just gets pumped back out by the sodium-potassium pump back down to resting potential. If you have a little more voltage, maybe 2 or 3 channels open up, a little more sodium comes in, but you still don't reach the threshold here or even here, it's still just a local potential. Remember that the action potential is all or none, which means it has to reach the threshold in order to set off the action potential. And if it does reach the threshold, even if it just barely touches the threshold, or if it goes way beyond, then every single time, 100% of the time, you're going to have an action potential. The reason for this is because once it reaches the threshold, it opens all of the voltage-gated sodium channels on the cell, and sodium can rush in at 500 times its normal amount. So as long as you hit the threshold here, then you get this spike of voltage that goes not just less negative, but actually becomes positive for a short time. And at the top of the action potential, what's happening is once it crosses 0, potassium channels are opening, but slowly. So still, it's only sodium coming in. And once sodium reaches its limit here at the peak of the action potential, about 30 millivolts or so, then those sodium channels inactivate. Now, sodium channels open really fast, close really fast as well. So the only thing that happens here is the voltage is going back down. And why is that? It's because at the peak of the action potential, the potassium channels are fully open. And so since sodium is cut off, the only thing that can happen is the voltage going back down because positive ions of potassium are going out of the cell, making the cell more negative. As it goes back down past 0 into negative range again, around the threshold right here is where the sodium channels reset, which means they can open, but they don't necessarily open at this point. They're able to, they reset. And here at the threshold level, as it's going back down towards resting potential, is where the potassium channels start to close. But again, you still see potassium coming out, not just to resting potential, but below resting potential here, because those sodium channels are closing very slowly. At this last point down here, what we have is what we call the undershoot or hyperpolarization phase, because the voltage goes slightly below resting potential as the potassium channels are closing. And then once they close, the sodium-potassium pump brings everything back to resting potential. So that is the review of what we already did. And for some reason, this PowerPoint format seems to have a problem with me just hitting the arrow button and cycling through. So bear with me here. It seems to be very slow. This is a problem I've had with these PowerPoint slides for a while now, and maybe I need to figure out a better way to approach it. Not really sure how to fix that, but we will figure it out. So bear with me. Trying to go back up. Let's see. So this is the last slide that we saw in the last lecture, where it's in a much better frame, though. It was very small in the last one. So again, the action potential movement down the axon is not a flow of ions, but it's a chain of events. So basically, each time that it goes from bare spot to bare spot is called saltatory conduction. And it basically, like a phone, it recharges itself at every bare spot along the axon. So I'll explain that in a little more detail. The local potential is a graded potential. So graded potential and local potential are basically the same term. You can interchange them. But the action potential, of course, follows that all or none law. So let's see if we can actually get this to change. It seems to be very stubborn, so I apologize for that. Let's go ahead and talk about something called the refractory period. So, one of the things that we talked about happening, or I talked about happening at the peak of the action potential, and I just went over it again really quick, is that at the peak of the action potential, one of the things that's happening is that sodium channels are becoming inactive. And if sodium is not allowed to come into the cell or move, then there is no way you can get another action potential. This is sort of one of the ways that the brain regulates itself, because we can't have too many action potentials at one time, or there might be chaos. So this is what's known as the absolute refractory period. At the point where the sodium channels are inactive completely, you have what's called the absolute refractory period. Absolute meaning, or at least this is my interpretation, there is absolutely no chance to get another action potential. You're already above the threshold still, and sodium channels are inactive. So from the peak of the action potential about to the time when you get down towards the threshold area, when potassium channels are starting to close, that is our absolute refractory period. Now, once the voltage goes down towards the threshold, one of the things I just talked about, and I talked about in the last lecture, is that the sodium channels reset. I did say that they don't necessarily open again, but they reset, which means that if the proper stimulation comes in, or if the channels are opened again, sodium can come in like it did before. Now, one of the things that's happening at the same time is that potassium is still flowing out of the cell because potassium channels are not closed. And at the time that the sodium channels reset, potassium channels are just starting to close. So not only are we going back down towards resting potential, but in the undershoot, or the hyperpolarization phase, you're actually a few millivolts below resting potential. So because sodium channels are reset and active and able to open, you can get another action potential during this portion, But because the voltage is still going back down, and because it goes below the resting potential, you need more sodium and more depolarization than you would when it's at rest. In this case, to go from, let's just say, -70 to -60 millivolts, you only need 10 millivolts of depolarization. When you're in the undershoot phase, the voltage may be down around -75 or even -80 millivolts. So if we think about it in those terms, you can get another action potential, but you need twice as much depolarization to get there. So that makes it relatively harder. So that's what we call the relative refractory period. In this area, in this portion in green here, as the action potential is going back down and hitting its last phases, past threshold and even past resting potential, you can get another action potential if you get a bunch of stimulation, if you get a bunch of sodium, a big depolarization. But it takes double the effort to do that. So during the absolute refractory period, this is the period where the sodium channels are inactive, and there's absolutely no chance of getting another action potential. Once the sodium channels reset, as the voltage is still going down and as the potassium channels are slowly closing, and you get to the undershoot phase, you can get another action potential, but it's relatively harder. So we call that the relative refractory period. Yes, you can get another action potential, but it would take double the amount of depolarization, double the input from sodium to do that. So those are the two refractory periods. And what that does is kind of ensure that your cells are firing at a normal rate. They don't fire too many action potentials and basically poop out or die, get too tired, and things like that. Really hoping that this works, but this slideshow does not seem to be that compatible with my computer. So we're just going to have to do it this way. My apologies. One day I'll get this fixed. So when we look at the things that can affect the action potential, and we can look at chemicals and toxins in the environment, we call them neurotoxins. Toxic being bad for your neurons. There are certain things in nature that can have an effect by affecting, they can have an effect on these action potentials simply by affecting how these channels open and close, or they can cause interference with those. So a neurotoxin is defined as a chemical or a thing that affects ion channels involved in the action potential. You may have heard of TTX, or tetrodotoxin. This tetrodotoxin works by blocking sodium channels. So what happens if you block sodium channels? Well, whether you guessed it or not, because I'd asked this in class, blocking sodium channels means that they can't open, that sodium can't come in, which renders that cell basically useless, and it can also cause the cell to die off. What is tetrodotoxin, or where does it come from, you ask? Well, it comes from the pufferfish. It is a toxin that the pufferfish gives off to hurt its enemies, something that was made in nature. And apparently the pufferfish is a very big delicacy in Japan as sushi. But if you don't know where the poison is, and you're not specifically trained for it, you have to eat it at your own risk, because it could not only kill neurons, but it could kill you. Now, being in my 40s, early 40s, the Simpsons came on when I was about 9 or 10 years old, and so I've been a fan since the very start. And I believe it was in season 1, maybe season 2, but most likely season 1, where Homer Simpson goes to a sushi restaurant and eats the pufferfish and thinks he's going to die. And first of all, he's a cartoon, and there have been many seasons of The Simpsons since. But so I don't know if 30 years is, if I have to say spoiler alert, but at the end he does not die. You can go watch this. It's on Disney Plus, by the way. So why does it kill you? Because it blocks sodium channels and doesn't allow the neurons to communicate with the body. And as the neurons die, so do you. Scorpion venom is something that is a little too close to my family. My brother-in-law, who lives in Arizona, was actually stung by a scorpion. And what scorpion venom does is, instead of blocking sodium channels so they can't come through, it actually opens sodium channels and prolongs the action potential. So if you think about chemicals that make your muscles move, if you keep the channels open, it's essentially going to keep that action potential going and, in essence, paralyzing you. So my brother-in-law was bit in the foot or the leg. Not bit, but stung. And he couldn't move that leg for at least 48 to 72 hours. So it basically, the scorpion paralyzes its victim by doing that, and it can escape or dominate or whatever it needs to do. Obviously, scorpions aren't going to dominate humans, but they can render them paralyzed temporarily. So that works by opening sodium channels and leaving them open. Other drugs can also affect ion channels like this. Local anesthetics like Novocaine, if you ever have a toothache and needed to get drilled or get a cavity, the doctor gives you an injection, dentist gives you an injection of a local anesthetic. He puts Novocaine in the gum right near the tooth, and what it does is it blocks sodium channels, and pain is actually a chemical messenger. So it blocks the pain channels so that he can drill or scrape or do any of those things, and you won't feel the pain. General anesthetics are ones that are used for surgery. It's usually a gas that you can breathe in, and basically it knocks you out long enough that you won't wake up during surgery, depending on how short or long the surgery is. And that works by opening potassium channels and keeping them open for the time that you are under. So anesthetics work by either blocking sodium channels or keeping potassium channels open. And that shuts down the entire central nervous system. We've even seen that ion channels can be modified by light or lasers. We don't have to go into that specifically. I'm already giving you a lot of information, so I'm not going to go into that nitty-gritty detail. And of course, no activity on the arrows, so we'll do it the old-fashioned way. Again, my apologies. So I had made mention early on that the axons are covered in this white fatty substance called myelin. And myelin is made by these helper cells called the glial cells. It's a fatty tissue that surrounds the axons and provides insulation and support. So in the central nervous system, which is the brain and spinal cord, the type of glial cell that makes the myelin for these axons is called the oligodendrocyte. In the peripheral system, as you'll learn when we go back to chapter 3, because we're skipping to chapter 5 after this, is called the Schwann cell. And that's just the neurons that extend towards the muscles and things outside of the brain and spinal cord. So our brain is very efficient. It has to be, because the brain controls every single thing that we do, every thought, every urge, every itch, every spontaneous movement, every planned movement, everything. So we need that signal to be just as strong at the end, so it releases the chemicals, as it was at the beginning. And some axons, like I talked about before, are very, very long. Some can be very short. So we need some sort of backup plan to ensure that the signal stays as strong no matter how short or long the axon is. And in this case, that's where the myelin comes in. It increases the conduction speed of the electrical impulse from 1 meter per second up to 120 times more, to over 120 meters per second. So it can make that signal travel much, much faster down the axons. So it gets there in milliseconds. The myelin, as I pointed out before, is not completely covered like a telephone cord or a charger cord, but actually kind of looks like little hot dog links where there are little gaps of bare axon called nodes of Ranvier, because Ranvier was the one that the researcher that discovered this. So there are little gaps or junctions in the myelin, so you're not just going straight through like, again, the cord of a computer or a charger. And the reason for this is that it allows, at that bare spot, it allows the charge to recharge itself and make sure that it remains just as strong at the end as it was at the beginning. So it basically jumps between the nodes under the myelin, and it jumps from node to node to node. And every time that it hits a bare part of the axon, it recharges itself. And what that does is, even if you are Shaquille O'Neal and the axon is going from your spinal cord all the way down to your little toe, and you're a big behemoth of a man, then the signal can stay just as strong to your toes as it was when it started. And this action potential jumping from node to node to node and recharging itself at every little bare spot is called saltatory conduction. Here's where I'm going to talk to you about the first behavioral psychological disorder before we even get to the last chapter where all the others are. If you've ever seen anybody or known anybody that's been diagnosed with MS, and you see that a lot of them are in a wheelchair, MS stands for multiple sclerosis. And multiple sclerosis is actually a disease in the brain where the myelin is destroyed. So the opposite of what the oligodendrocyte does. Instead of making myelin for the axons, myelin gets stripped from the axons. And therefore, you reduce the conduction speed, you reduce the efficiency. One of the great things about myelin is not only that it increases the speed up to 120 times, but that the signal travels down in one direction nice and strong, recharging itself along the way. If you don't have this myelin to do that, then the information could go forward towards the axon terminal, but it can also go out the sides, it can propagate backwards, and the signal just might not get there. So a lot of this information, which is motor in nature, going to the muscles, this is why people are wheelchair bound, because those signals are not getting there. And if the signal doesn't get there, then the neurotransmitter, the chemicals, are not released and nothing happens. So a lot of times people with MS, I actually have a neighbor on my street who has MS, and he's been in a wheelchair for pretty much as long as I've known him. Maybe not completely, but it's been a while. So it first starts attacking the muscles because the signal doesn't get there, and it also eventually affects learning, memory, and cognitive processes, right? Thinking, because the connections within the brain will start to slow down without white matter, without this myelin. So multiple sclerosis is not fun, and now you know about how it works in the brain. Trying my best to not do this manually, but alas, it fails. So here is a picture of the myelin, and you can see that it is layered almost like insulation in your attic on the axons where there are bare spots here and here. You can see, it's more like hot dog links, that there's bare spots here. Here's the oligodendrocytes, which means it's in the central nervous system. Here, Schwann cells are here, so this must be in the peripheral nervous system. And you can see the gaps in between here. So the node jumps through from here to here to here to here, and it recharges itself at every little bare spot. This ensures that the signal is just as strong at the end when it needs to do its thing as it was at the beginning when it was created. And this is sort of the brain's foolproof way of ensuring that the signals that we get from the neurons go to where they need to go and that they're nice and strong and efficient. I'm not even going to try it this time. I'm just going to go ahead and go out and come back in. Maybe you're asking yourself, do we need the slide full screen? I like it that way. So here you can see glial cells. Glial cells are the helper cells. So our brain is actually 90% glial cells, 10% neurons, and those glial cells are doing everything they can to help the nervous system to be as efficient as possible, because that's what helps us to think, feel, act, survive, move, right? All of those things. So here you can see that glial cells are present. They're the little bumps, and you can see some of these connections between neurons. The more glial cells there are, the more connections you see. When we're learning things and coordinating and figuring things out, we need these connections to be nice and strong. So let's move on a little farther down the line. And again, glial cells do pretty much everything. So the answer is all of the above. Glial cells are helper cells. They migrate. When neurons are migrating, they help to put them in their final spot, so they're nice and organized in columns and rows. They respond to injury and disease by removing debris and bad things. Any cells that die off get eaten away and flushed out with the help of glial cells. Nutrients and energy are provided by glial cells. So what do they do to help out? They give myelin. They move neurons to where they need to go. They nurse them back to health. They eat away at the bad stuff and help the good stuff. So they do basically everything. Scaffolding for migrating neurons, which means they provide a passageway to get them to the right spot. Respond to injury, remove debris, nurse them back to health, provide energy, and we see 7 times more neural connections when glia are present. So glial cells are the ultimate helper cells. Specific ones may have specific jobs, but overall, they are there to make sure your nervous system is running as efficient as humanly possible. So you can see the astrocytes are the ones that help the neurons and give them energy and nurse them back. You also see more astrocytes depending on the complexity of this brain. So looking at the leech brain, the frog brain, the mouse brain, yes, they have certain movements that they have to do, but they're not very complex in their behaviors. So here a leech has almost 0 astrocytes per neuron. A frog has about 0.3. So basically 1 astrocyte for every 3 neurons. In the mouse, you see about 0.4. In a rat, about half. So 1 astrocyte per 2 neurons. In a cat, which has more complex behavior and can do a lot more things, you see 1.1. So a little more than 1 astrocyte per neuron. And in a human, we see about a 1.5 astrocytes per neuron. So it's a much bigger ratio. Much bigger ratio in humans, because humans can not only move in much different ways, but also for thinking and reacting to the environment. We can plan ahead. We can think about things. We can make decisions based on information. So these astrocytes are going to be more numerous in the more complex human brain. You might see something similar in a ape or a monkey brain. Very similar to us. So again, I'm not screwing around. I'm going to just do it this way for the rest of this presentation, if you don't mind. Not that you can stop me. And we'll move on from there. So what happens now when the signal reaches the axon terminals? Well, here you see the axon terminal. This is a blown up picture here. Here's 1 neuron, and here's the axon that is myelinated all the way down, and it ends in these terminal buttons or synaptic terminals. And here you see the dendrites of the neighboring cell. So at some point, the signal has to get from this cell, which we call the presynaptic cell, pre meaning before the synapse, to the postsynaptic cell, which is here, postsynaptic neuron, after the synapse. And the synapse, as mentioned earlier, is the gap between the two. So again, originally, hundreds of years ago, or at least 100 or a couple hundred years ago, it was believed that the electrical activity was all that we needed. And that the neurons were actually connected like wires in a circuit, which turns on your lights in the house and things like that with the flip of a switch. But what was discovered more recently, maybe about 100 years ago, is that the electrical activity was not the whole story, and especially here, that they are not connected. Here you see this synaptic gap or synaptic cleft, or sometimes it's just called the synapse, between the two cells. The sending cell is going to release something that is picked up by the next one. As we know now, these are what we call neurotransmitters, or the chemicals in our brain that facilitate the communication. So the electrical signal comes down into the presynaptic terminal. Something happens that causes the chemicals or neurotransmitters to be released. They swim through this little synaptic cleft or synapse, and they bind to these receptors like a lock and a key on the postsynaptic cell. So let's use a chemical that you guys are probably familiar with called dopamine. And we'll just say that signal comes in, dopamine is here at the bottom of the axon terminal. It gets released into the synaptic cleft or gap or the synapse, and it is picked up by dopamine receptors on the postsynaptic cell. So they fit together like a lock and a key. Dopamine will only bind to and open dopamine receptors. Acetylcholine will only work with acetylcholine receptors, glutamate with glutamate receptors, etc. So how did we come across this, and how did we learn to understand that chemicals are the actual messenger? That doesn't mean that the electricity is not super important, because the electrical activity is what causes these chemicals to be released to the next cell or to a muscle or to another structure in the brain or in the nervous system. So let's go ahead and figure out or take a look at how this was discovered. So about 100 years ago or so, give or take, of course, there was a German scientist, and his name was Otto Lowy. And Otto Lowy was looking at frog hearts. And he had one frog heart in a jar, like a jar of, like a beaker, and still had some of the nerves connected to it. Here, we see something called the vagus nerve. I know it's not spelled like Las Vegas, but it's the vagus nerve, VAGUS. And here I go, giving you another joke that I don't apologize for. What happens in the vagus nerve stays in the vagus nerve. So the heart, in order to survive in the beaker, had to be in a solution. Maybe salt water had some stuff in it, so it was like taking a bath. And there were actually two of these nerves that came from it. One was called an accelerator nerve, which would do exactly what you think it would do. It would stimulate the heartbeat and make it go faster. The vagus nerve is actually stimulated to slow down heartbeat. So if your heart's beating too fast, the signal from the vagus nerve would cause it to slow down. So here's the frog heart in jar A, and here's the frog heart in jar B. What happened was Lowy wanted to see exactly what would happen if he used electricity. So you can stick an electrode, which gives off electricity, and stimulate one of these nerves, either the accelerator nerve or, as pictured here, the vagus nerve. So what he did was, in heart A, in beaker A, he had stimulated the vagus nerve, which, again, without stimulation, it was beating pretty fast. When he stimulated this nerve electrically, it caused the heartbeat to slow down. When he stimulated the accelerator nerve on the other side, what happened? It did the opposite. It sped the heart up. So there we go. We can see that giving electrical activity is going to cause that heart to speed up or slow down based on electrical activity. So what was discovered before this seemed to be true. Okay, well, then Lowy said, what if I took some of the solution in a beaker from heart A when it was accelerated or when it was slowed down, and I just poured some of that solution into beaker B, where heart B was. So there was no electrical stimulation here. Once Otto Louis hit the vagus nerve and the heart started slowing down, he took a little of that soup, a little of the bath that the heart was laying in A, in a beaker, and he poured it into B. And guess what happened? That heart in B changed to match heart A exactly in the similar way. Stimulate the vagus nerve, take some soup, put it into the next jar, and that heart also slowed down. Stimulate the accelerator nerve to speed the heart back up, take some of that solution, that soup, put it into heart B, and that also sped up heart B. So Lowy came to the conclusion that yes, the electrical activity is necessary, but it is not what's sending the message. The message is being sent by chemicals, not action potentials, to be able to change the heart rate. And that's how we discovered that the brain has chemicals, and it's those chemicals that are responsible for the communication in the nervous system. Actually it did work here. Look at that. So let's see if I can. Of course, now it starts working when I do this. Coming back into it. So here, and you can, by the way, click on. Well, you can't click on this link, I guess, but you can because you have access to the slides. So when you have access to the slides, and maybe you're following along as you go through this lecture, you can simply click on this link, and it'll show you the animation here. But essentially, what happens is, here is the, this is a computer simulation. The action potential here that we see from the voltage, right, comes down the axon and enters the axon terminal. In the axon terminal, this is where the neurotransmitters, the chemicals, are actually made. And they're actually stored in these little tiny sacs, these little tiny bags almost, called vesicles. Vesicles, vesicles. The vesicles are here in the bottom, but they are not going anywhere. They're closed bags, right? Closed sacs of neurotransmitter. The little red dots are meant to be the neurotransmitter. So when the action potential comes in and the electrical activity comes in, how does it actually cause the chemical to be released? And then what happens to the chemicals? Well, here we see that there is a little receptor channel here on the end of the axon terminal. Now, this is not a sodium channel. This is not a potassium channel. This, by the way, if you don't know what Ca2 plus is, or Ca plus plus, is something that our body needs. And in milk commercials, we know that it does a body good because of this. This is calcium. So there are voltage-gated calcium channels on the axon terminal. When the action potential, the electrical signal, comes down, and again, it has to be just as strong at the end here as it was at the beginning, right? This is sort of our foolproof way of making sure that our nervous system is working. When the action potential comes down, it depolarizes the membrane and opens the voltage-gated calcium channels. Just like reaching threshold opened the voltage-gated sodium channels, and that rush of sodium opened the voltage-gated potassium channels, here, down here in the axon terminal, the electrical signal opens up calcium channels, and calcium comes into the terminal. When the calcium comes into the cell, then these vesicles bind to, as you can see here with this one, bind to the edge, the membrane of the axon terminal, and it opens up the axon terminal, opens up the vesicles, and the neurotransmitter is released into this cleft. It's released into the synapse. Once that happens, then the neurotransmitter has to swim and find its way to this postsynaptic cell where these little 4 little rectangles are the receptors. If this is dopamine and these are dopamine receptors, dopamine will then bind to the dopamine receptors. When the receptors are bound to, it causes them to open ion channels. The most likely thing that happens is it opens sodium channels. If sodium channels are opened on the next cell, that's right, sodium can come in and produce what we call an EPSP. So since this is the postsynaptic cell, any change in voltage is called a PSP, postsynaptic potential. Now, obviously, we have 2 things that can happen. It can either cause a depolarization, which is excitatory, so we call it an excitatory postsynaptic potential, or it can cause potassium channels to open and potassium to go out of the cell, or chloride channels to come into the cell instead of sodium, and that makes the cell less likely to fire. That actually hyperpolarizes the cell and makes it more negative. In that sense, we have what we call an IPSP. Instead of excitatory, it's inhibitory postsynaptic potential. So I don't know if the animation will play, but we're going to try and see if it'll work. No, nothing happening. Nope, whoops, and now it's going too far. So temperamental. So, this is where we were. So the 3 steps is that the action potential comes down, opens voltage-gated calcium channels, calcium comes into the cell. Once calcium comes in, it causes the vesicles to bind to the membrane and to release into the synapse. Once that's number 2. So once the neurotransmitter, once the chemicals swim down through the synapse, they bind to the postsynaptic receptors, and that causes either an EPSP, most likely with sodium coming in, making the cell a little more likely to fire, a local potential, or chloride can come in or potassium can come out, and that can cause an IPSP, or an inhibitory postsynaptic potential. So, action potential depolarizes the membrane, calcium channels open, calcium comes in, neurotransmitter is released into the cleft or the synapse. It binds to the receptors, and that causes these ionotropic receptors, which means that they just control ion channels, ionotropic, to either change the potential in a positive way by depolarizing, and you get an EPSP, or by causing the cell to be less likely to fire, and hyperpolarizing, which is called an IPSP because it's inhibitory. So the ionotropic receptors are the main type, but they're not the only type of cells. Ionotropic are very fast acting because all they do is open channels and close channels. So what that does, as you saw with sodium and potassium, although potassium takes a little longer to open, is a very direct and rapid effect. Boom, the channels are open. Boom, sodium comes in. Boom, the channels are open. Boom, chloride comes in. If sodium's coming in, you're getting closer to an action potential with an EPSP. If chloride's coming in, you get an IPSP. But there are other types of receptors too that may open ion channels indirectly later on, and they produce slower but longer acting effects. So it can be something like eventually opening channels, but they can cause other things to happen inside the cell. They can cause enzymes to break down things. They can cause proteins to be structured. We don't need to get into the specifics because I've already given you a lot of little details. But what you need to know about metabotropic receptors is they can cause other things to happen other than opening ion channels, although eventually they can open ion channels. And their effects are much slower, but much longer lasting. So making proteins, activating enzymes or things like that, right? You don't need to know these things specifically. Ionotropic, only opening and closing ion channels, very fast. They start, they stop really quickly. Metabotropic, very slowly, but much longer lasting. So we know that this transmission of chemicals is much slower than electrical transmission, but that's where the actual message gets across. And this is where the actual message has trouble getting across in people with MS. Oh, now it's working. Hallelujah. So when you activate the receptors on the postsynaptic cell, as I mentioned, there are two possible effects: hypopolarization or depolarization, which causes an excitatory signal because it makes the cell less negative or more positive, and we call that an excitatory postsynaptic potential. Majority of the time, it's going to be sodium channels that open because we want the signal to go across, we want the cells to fire, we want that signal to get there and the chemicals to work. So in EPSP, open sodium channels. If enough sodium channels are opened and the depolarization is enough to reach the threshold, then that next cell gets the action potential and starts all over again. However, so it makes the cell or neuron more likely to fire. However, there are instances where we don't want the cells to fire as much. Too much electrical activity actually can be harmful. So one of the things that too much electrical activity in the brain does is causes anxiety, it causes insomnia, and in extreme cases, it causes seizures. If you've ever seen somebody have a seizure, whether on TV or in real life, a lot of times they're on the ground shaking as if they're being electrocuted. And that's not far from the truth, because too much electrical activity, your brain is literally being shocked. So it causes those convulsions. Because of these things, if you want to calm your brain down because of anxiety, epilepsy, insomnia, you need chemicals to make you calm down and even sleepy. So if your brain feels that there's too much activity, then it's going to cause the cells to be less likely to fire action potentials in order to calm itself down, or certain medications can do this. That's where you would cause an IPSP. So an IPSP either opens potassium channels, so potassium comes out of the cell, making it more negative, or it opens chloride channels and chloride comes in the cell, which is negative, or maybe it's a combination of both. This combination of an IPSP causes hyperpolarization, causes the cell to go below resting potential, and it makes the cell less likely to have an action potential and less likely to fire, because maybe we need to calm our system down. So, both EPSPs and IPSPs are graded potentials. And in order to get them to reach the threshold, remember the first slide from this one, I showed a bunch of little local potentials had to kind of combine in order to reach the threshold. So these graded potentials on their own are just going to be pumped back out, and you're not going to get the cell to fire. We need two things to happen. First of all, we need all of these things to happen at the same time or within a very short time. Otherwise, you're just going to get little waves that go up, down, up, down, up, down. So they have to happen at the same time, and if you need enough sodium channels open, you probably need different inputs in different locations. So accumulating over a short time is what we call temporal summation, as in time, that they happen really quick. Combining inputs from different locations, remember from the pictures that there are a lot of axon terminals that can release chemicals, and there's a lot of dendrites that can pick up chemicals on the same neuron. So we need many inputs from different locations, so many inputs of sodium channels opening on this cell, and we need them to do it at about the same time. If you have the combining the different inputs is called spatial summation. So you need both spatial and temporal summation in order to make the cell fire, and I'll explain how that works. So even if you have a bunch of targets, a bunch of sodium channels that open, if they don't open at the same time, you might get something like this, where you have wave, 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 but not enough for it to combine to be able to reach the threshold. If you have one neuron or one sodium channel opening, you have this, and then it goes back down. If you have a couple of them, you can get it and they're at the same time, then it goes a little higher before it goes back down. If you have 3 or 4 or more at the same time, then it builds upon one another, and if it hits the threshold, boom, you have your action potential. So in this case, the neuron acts as an information integrator because some of the signals will be EPSPs, causing sodium to come in. Some of them may be IPSPs at the same time. So our brain, our neurons have to integrate that information, and if you get more excitatory than inhibitory, kind of like doing math, then they combine algebraically, and if it reaches the threshold, you have that all or none process, so it will always fire as long as it reaches the threshold. If it never reaches the threshold, even if it's just below the threshold, those voltage-gated sodium channels don't open, and you do not get an action potential. That's why this is an all or none phenomenon. So here is where you'd see temporal and spatial summation. I apologize that the pictures don't show up on this slideshow on my computer, but you can go to the pages where I have this written out, and you can see those pictures. But basically, what it says is, right, temporal summation is that they come in at the same time. Spatial summation is that you get enough channels to open to give you enough depolarization at the same time to reach the threshold and cause another action potential to fire. So, one of the things that we've been talking about, that I've been mentioning, is the sort of fail-safe or foolproof plans that the neurons have. The myelin is number one because it makes sure that the signal is just as strong at the end as it was at the beginning to ensure that pretty much every time you're going to fire an action potential, and the signal is going to be strong enough to release the chemicals. When we release chemicals, another fail-safe of our nervous system is that we generally release more neurotransmitters than we need just in case. The receptors can only pick up a certain amount, but we want to know that there's enough for them to pick it up. If we have too little of a neurotransmitter like serotonin, then we could have problems. Depression, by the way, is biologically seen as one of the symptoms is a lack of serotonin. That's why antidepressants come about. Antidepressants are normally targeted for serotonin either specifically or as a package. And if you increase the amount of serotonin, chemically, you are balancing off your chemicals in your brain. You may have heard the term chemical imbalance. That just means there's less serotonin than some of the other neurotransmitters or chemicals. And so that's what medicines or drugs like that do. So when neurotransmitters are fired into the synapse, we usually have more than we can pick up, and that's just in case. But that may be a problem if we didn't have any way to take care of it. So there are two ways that neurotransmitters are removed from the synapse in order to make everything nice and clear. The first one is called reuptake. And basically, the neurotransmitters are, because it would be a gigantic waste of neurotransmitters if they were just thrown out, when it is determined that the neurotransmitters have done their thing, the original presynaptic cell that released the neurotransmitters can actually suck them back up and recycle them and repackage them back in the terminals. So if you're wondering how long man has known about recycling, well, your brain has been doing it since the beginning. Very efficient, very environmentally friendly. So this process of reuptake is basically the extra neurotransmitter being sucked back up into the presynaptic cell and repackaged to be used again later. The other way that neurotransmitters are removed from the synapse is that enzymes can come in and eat away at them and break them down to be flushed out. So that's called enzyme inactivation or enzyme degradation. Enzymes break down the neurotransmitter in the cleft, and now there's room for more new ones. So the reason, by the way, that I skipped from chapter 2 to chapter 5, so after this chapter is done, the next one will be chapter 5 on drugs and their effects on the nervous system, is because when we talk about neural communication, we talk about the specific chemicals that flow in your system. And what drugs do, and by the way, a drug doesn't just mean marijuana or something like that. A drug is a medication. It is basically anything that is not made inside the body that changes the way the body reacts. So the two main ways that drugs work is they cause more effect of a neurotransmitter, which could be two things. It could be more neurotransmitter released from the presynaptic cell, or it could be more neurotransmitter that's taken up by the next cells. Majority of the time, it's more that's taken up by the next cells in order to increase the flow, in order to increase transmission. Sometimes we may have too much of a neurotransmitter, as we'll get into with schizophrenia. In a part of the brain called the basal ganglia, we have too much dopamine, which is actually responsible for some of those psychotic symptoms that we'll talk about at the end of class. In that way, we need to reduce the amount of neurotransmitter. So, some of the drugs actually either partially or fully block the receptors from getting the neurotransmitter, or in rare cases, they can cause less of the neurotransmitter to even be released from the presynaptic cell. So the drugs that the drugs that cause more activity of a neurotransmitter are called agonists, and an agonist can be in three different ways. An agonist can cause more neurotransmitter to be released. It can mimic natural transmitters to fit in the receptor. I like to use the term skeleton key, but I was told by a colleague of mine that skeleton key is kind of an outdated word, so maybe a better term would be master key. So some drugs are basically like a master key, and they fit into the receptor, lock and key, keeping it open longer and allowing more of the neurotransmitter to come up. So if your serotonin receptors or your dopamine receptors are not picking up very much and you need more serotonin in your system, then this drug may cause the receptors to stay open longer and increase the amount of neurotransmitter, the amount of serotonin that you're getting. There is also a third way that agonists can work, and that would be to either block the reuptake or block the inactivation, therefore not keeping the receptors open longer, but keeping them in the synapse longer. Then when your receptors reset and are ready for more, you have that extra neurotransmitter still there that they can take up. That is exactly how SSRIs work. So if you've ever heard of Prozac or Zoloft, there's a lot of commercials around for them. And if you've ever watched the commercials and heard the term SSRI, that stands for selective serotonin reuptake inhibitor. So basically this idea of reuptake, where we're recycling the neurotransmitters, they're blocking, right? They're inhibiting the reuptake. So they're blocking the neurotransmitter from being recycled. What does this do? And by the way, SS, selective serotonin, just means that it's specifically serotonin that is being blocked, or that's reuptake is being blocked. So by doing this, they leave more of that. It's not recycled. It's left in the synapse. And then when the receptors are ready, they'll take up the rest because it's still there. So you have the lock and the key. The lock is ready. There's the neurotransmitter in there to regulate. So this will increase the amount of serotonin that flows through your brain, that flows through your neurons. There's also ones that instead of preventing reuptake, they prevent the inactivation. So MAO inhibitors are blocking the enzymes that break down the neurotransmitter. So maybe some gets recycled, but the ones that get inactivated, they block the enzymes from doing that, and you get the same effect. It is leaving more neurotransmitter in the synapse, and when those receptors are ready, they'll take up the rest. So you may have heard the term SSRI. You may have heard the term MAO inhibitor. And MAO, by the way, stands for monoamine oxidase, which is the enzyme that breaks down dopamine, serotonin, and another chemical we call norepinephrine. So this won't necessarily be on the exam, but at least now you know how chemically these drugs called antidepressants may work. So when we try to regulate synaptic activity, of course, the brain tries to regulate itself. Agonists can work by either causing more release of a neurotransmitter, but usually by either mimicking a neurotransmitter and leaving the receptors open longer, or, like I just said, blocking reuptake or blocking enzymes from breaking down the neurotransmitter. Either way, this activity is agonistic. It is allowing for more neurotransmitter to go through your system, your nervous system. So, antagonists work by either releasing less neurotransmitter. As you see here, presynaptic inhibition decreases the amount of neurotransmitter released, and that can cause less to be picked up, or majority of the time with antagonists, it either partially or fully blocks the receptor, kind of like sticking gum in your lock so you can't put the key in, right? Either partially or fully. So we can decrease the amount of neurotransmitter released, or we can increase the amount of neurotransmitter released. How does that work? Well, we know that calcium comes in and causes the neurotransmitter to be released. So if we can affect how much calcium comes in, we can affect how much of the neurotransmitter is released. The more calcium that comes in, the more neurotransmitter will be released. The less amount of calcium that comes in, you guessed it, the less amount of neurotransmitter that's released. And in addition to these calcium receptors or calcium channels on the cell, we have what are called autoreceptors. The autoreceptors are the ones that bring back the reuptake of the neurotransmitter, of the chemicals. So they basically sense the amount of neurotransmitter that's left over, and they cause the neuron to open those channels and either reduce the output or suck back up whatever was already released. And of course, helping the neurons to do this, they don't function on their own. We have those big helper cells, glial cells. They prevent the neurotransmitter from spreading to other synapses, so they keep it local in the synapse. They don't let it trickle out. They can absorb and help to recycle the neurotransmitter, so glial cells are helping with that reuptake process. And they may even release their own glutamate to regulate the presynaptic release. So they're helping this whole process. So now you know the electrical process and the phases of the action potential, and now you know how the chemicals are released once that electrical signal gets there, and what happens to the next cell. We've even seen how we can cause more or less activity of a neurotransmitter using drugs and with the help of glial cells. So, wow, this looks very interesting that's all together like this. So I'm going to go ahead and put it down here. Sorry, I don't like all this, all this interference. Let's put this down a little bit. And let's go back to present. We are almost done here, I promise. So, we talked about a few of the neurotransmitters, but I'm going to go ahead and look at some of the actions, and we'll talk about some of these neurotransmitters and who they are, and what they do. Some of them have very specific effects on behavior. Some of them have sort of general effects. So, in order to understand this, we also have to understand that the receptor types are like a lock and a key, and there may be more than one type. So dopamine actually has 5 different receptors for dopamine, and they're just simply labeled D1 through D5. So D1, D2, D3, D4, D5. And certain types of dopamine may only bind to certain of those receptors. Acetylcholine is the neurotransmitter that is sent to the muscles to tell them what to do. Acetylcholine is also within just the brain itself and is actually implicated in, those are the neurons that are destroyed in Alzheimer's disease, as we'll get to in chapter 12. So acetylcholine has 2 different receptor subtypes. One is called nicotinic. I know this says nicotine, but that's where nicotine actually got its name. The ingredient in cigarettes that stimulates the body. Now, I've never had a cigarette, but I know that when people smoke, they also, not only are they stimulated in the brain, but seem to get a little bit of almost a tingling sensation maybe in the muscles, and this is why. It's the nicotinic receptors. But acetylcholine also has muscarinic receptors. So we have also seen that neurons can release more than one chemical. It used to be believed that only one chemical could be released in neurons, but as technology gets better, we have seen that certain neurons can release more than one type of cell. So there used to be a law called Dale's Law, which was a guy named Dale, who said and claimed that neurons can only release one type of neurotransmitter. We know that that's not true now. Some are fast acting and some are slower acting. Some are inhibitory, some are excitatory. What we now know through the modern marvel of technology is that neurons can release more than one chemical. One can be fast acting and one can be more slow acting. A neuropeptide may be like substance P, which is for pain, as we'll go through here. You can have 2 or more fast-acting neurotransmitters. We can even have some that are excitatory that cause EPSPs and some that are inhibitory that cause IPSPs at different synapses from the same neuron. So this contradicts Dale's law, and now something that happens every single time is what's called a law. So that had to be downgraded to a principle because we know it does not happen all the time. So this contradicts Dale's principle. Sorry that you got downgraded, Dale. That neurons can only release one neurotransmitter, because that's not true. There may be some neurons that only release one type, but we know that a lot of them can release more than one type. So what are these neurotransmitters that your professor speaks of? Well, here we see that there are 4 or 5 specific neurotransmitters that have specific pathways and do specific things. And then we have 2 or 3 amino acid ones, which I call the global neurotransmitters, because they're located all over the brain and they're kind of globally working through the entire brain, not just specific pathways, in order to do things. So let's take a look at what these are. And then tables like this are always really good study tools to be able to make flashcards or get a really good summary of information. So the first neurotransmitter that I just talked about is called acetylcholine. That's up here at the top. Very specific pathway within the brain and spinal cord, in the CNS, or within the brain itself, I should say. And it's also the one that is involved in making the muscles do basically everything that they do. So acetylcholine to the muscles is what makes them move. So anytime you're moving your muscles, your brain secretes and your neurons secrete acetylcholine that's telling the muscles what to do: expand, contract, do things as simple as walking, or do things as complex as trying to hit a baseball at 90 miles an hour or more. In the brain, it's involved in learning, and with Alzheimer's disease, and people lose their memories, their ability to recognize things, to remember things, that's because these acetylcholine neurons are being destroyed. We also have the monoamines, which I talked about as serotonin, dopamine, norepinephrine, and sometimes we can consider epinephrine. So if you've heard EpiPen, I'm going to also tell you about something that is a little pet peeve of mine, but it's really not a big deal unless you're me and you get annoyed by it. Stay tuned for that. So serotonin is the one involved in mood, sleep, and arousal. It can be involved in aggression. It plays a huge role in depression and obsessive compulsive disorder, or OCD, and is even related to alcoholism. So some of the genes that code for serotonin, as we talked about genetics in chapter 1, can also code for your susceptibility to alcoholism. It also may be why a person that's depressed may turn to alcohol as a way to cope with it. So serotonin in lower levels is believed to cause, like I said, the chemically induced depression. There may be, of course, environmental things like losing a loved one, or failing a class, getting fired from a job, that can also contribute to that. And if you're wondering why serotonin, when we talked about the SSRIs, it's because serotonin seems to be the ringleader of these monoamines, and serotonin, when you regulate it, it seems to bring the others up to speed. I'm going to skip dopamine for a second. That's my favorite one, so I'm going to get back to that. And talk about norepinephrine. So norepinephrine in the brain is due to, or is involved in levels of arousal of our brain. So during stress, it's released, which causes our system to ramp up. When we're really awake and alert, we have more norepinephrine in our system. When we're really tired and sleepy and can't concentrate, we have less norepinephrine. So the original antidepressants actually targeted both norepinephrine and serotonin, because if you're depressed, not only is your mood low, but your energy levels are low. So your arousal level is low, which means your brain is functioning at a lower level. So it was serotonin and norepinephrine. So the original antidepressants actually targeted both of them. But the reason that serotonin and SSRIs seem to work a lot better is because when you're targeting one system, that's less side effects than targeting more than one system. And as I mentioned before, if serotonin is regulated, it seems to bring the others with it. So serotonin seems to be the ringleader of the monoamines. And if you level that off, the others will follow with it. Epinephrine is sort of one more sort of chemical synthesized from norepinephrine. It turns into epinephrine. It's more of a stress hormone that's related to it, but plays a minor role as a neurotransmitter in the brain. So the thing that annoys me about norepinephrine or epinephrine is these things, as you'll learn in chapter 3, are called something different when they're in the central nervous system or peripheral nervous system. That glial cell that makes the myelin in the central nervous system, brain and spinal cord, is called the oligodendrocyte. That same type of glial cell that's outside in the peripheral nervous system is called a Schwann cell. So you will see that there are different terms in chapter 3 for things in the CNS and in the PNS. In the CNS, central nervous system, we have epinephrine. The same epinephrine in the peripheral nervous system is actually a hormone, which is called adrenaline. Adrenaline and epinephrine are essentially the same thing, but inside or outside of the central nervous system. So the reason that I get a little bit annoyed is because if you've ever seen anybody that has allergic reactions that can cause what we call anaphylaxis or anaphylactic shock, which causes your throat to close up and the inability to breathe and could cause death, they carry around what's called EpiPens, which stands for epinephrine. The problem is it's not in the central nervous system. So you're not injecting it into the brain, which you probably couldn't get through the skull. You're not injecting it into somebody's spinal cord. You're injecting it into the peripheral nervous system outside, most likely in an arm or in the heart area, right? So in that case, is it really epinephrine? People go to great lengths to call that adrenaline. So for me, it just annoys me, and this is the sound of you guys collectively rolling your eyes at me, that it would be better suited to be called an adrenaline pen and not an epinephrine pen, because again, epinephrine specifically is related to the brain or the central nervous system. So that type of pen that can, or that injection thing that can essentially save someone's life, should be called an adrenaline pen, and yet we hear it as an epinephrine or an EpiPen. All right, rant over, sorry. The last of the monoamines here is called dopamine. Dopamine has a lot of different features. It controls to movement control, so it's a lack of dopamine in the same area where there may be too much dopamine in schizophrenia, which causes the lack of movement ability that you see in somebody with Parkinson's disease, like Michael J. Fox, who was the star of Back to the Future movies. It promotes the reinforcing effects of food, sex, and drugs. So anything that you can become addicted to. And we'll talk a lot about addiction in Chapter 5 coming up. So dopamine is the main neurotransmitter that's involved in the addiction pathway. And it doesn't just have to be drugs or alcohol. Addiction to gambling, addiction to the internet, addiction to video games, addiction to eating. All of this, addiction to sex, all of this is involved in the dopamine pathway. And of course, as I mentioned earlier, in schizophrenia, there's too much dopamine in one area of the brain. In Parkinson's disease, there's too little in that same area. So one of the things when scientists and psychologists were coming up with drugs to be able to treat schizophrenia and Parkinson's disease, agonists and antagonists, depending on what was going on, one of the things you had to watch out for was the side effects, because, or at least the dosage levels. Because if you had, let's say that you had schizophrenia and they gave you a dopamine antagonist, if you blocked the dopamine too much, you might actually get side effects called tardive dyskinesia, which could mimic the effects of Parkinson's disease, because what you effectively did was not regulate the dopamine, but maybe block too much, and now you have a lack of dopamine as opposed to too much, which is going to mimic the effects of Parkinson's disease. In the same breath, with Parkinson's disease, people gave L-Dopa, which converts to dopamine in your brain, and L-Dopa can cross this protective area called the blood-brain barrier. We'll get to that in Chapter 3. But people that had too high a dose of L-Dopa, guess what? Had some of the psychotic symptoms seen in schizophrenia. So there is a fine line between having too much, too little, and the right amount. Those are your specific neurotransmitters. Down here, these amino acid neurotransmitters, glutamate, gamma aminobutyric acid, better known as GABA. So please, we can always just use GABA. And to a smaller effect, glycine. We don't see glycine as much in the system. So the two main ones are glutamate and GABA. These are the ones that I call the global neurotransmitters. And by that, I mean that they are all over the brain, not in specific pathways like these, and they are looking at the overall activity of the brain. They also work together kind of like a scale, where if you have more glutamate, it's going to be less GABA. If you have more GABA, it's going to cause less glutamate. So it's like a yin-yang, or like the sides of a scale. More of one means less of the other. What are they? Well, glutamate is the main excitatory neurotransmitter in the brain, so it's going to cause EPSPs, and it's going to cause more activity in the brain and spinal cord. It is very important for learning and memory, and implicated in schizophrenia. When you have too much electrical activity in the brain, so like I said, our body likes to be balanced. Our neurons like to be at negative 70 at resting potential when nothing is happening. And unless it hits the threshold, it's even staying at negative 70 because of the sodium-potassium pump. Well, we need a certain level of arousal to be able to maintain our focus, to learn things, to concentrate on things. So we do need a good amount of glutamate to keep our brain running in a good fashion. But we also don't want too much activity because that could lead to those things I talked about earlier: seizures, anxiety, stress, right? Insomnia. So you don't want to have too much glutamate, or that can be really bad. You can also think of it like drinking coffee or an energy drink. You have one energy drink or one coffee, maybe that puts you, gets you out of being groggy and tired, and you're in a place where you can work or learn or both. But if you drink 3 cups of coffee, or you have, 3 Monsters or Red Bulls or whatever, then you're going to be, spinning your wheels and trying to bust through the wall like the Kool-Aid man, and that's not any good either. So we need GABA. Again, that's gamma-aminobutyric acid, or GABA. GABA is the predominant inhibitory neurotransmitter. What it does is causes IPSPs. Like I said, sometimes it's not good to have too much electrical activity in the brain for that reason. So what GABA does is it basically makes you sleepy and calm. So it's going to respond to alcohol. The more alcohol you drink, as we'll cover in chapter 5, the more sleepy you get. And taking tranquilizers, which we call benzodiazepines. So the more GABA you have, the more likely it is that you're going to fall asleep. And deficiency in GABA is one of the, or receptors, is one of the causes of epilepsy, which is seizures. So again, more GABA, less glutamate, makes you sleepy, shuts down the nervous system. More glutamate, less GABA, ramps up your system. Glycine is another inhibitory transmitter in the lower brain. And glycine also, like GABA, tends to calm you down if there's a little bit too much activity. If you've ever heard of the poison called strychnine, which causes convulsions and death, it's because what it does is it affects glycine activity and doesn't allow you to control that inhibitory portion of the brain. To make things even more complicated, you're welcome, we have things that are what are called neuropeptides, which are like also neurotransmitters. They're chemical messengers in the brain that lead to certain things. As I told you guys with substance P, that pain itself is a neurotransmitter or a neuropeptide. So when we are in pain, although pain sucks, we need it because it's our body's alarm system. So it allows us to let us know if something is wrong. If you sprain your ankle, you're going to feel a lot of pain in that ankle, and that tells you, hey, dummy, something's wrong with your ankle, stay off of it. When we are feeling really good, first of all, in response to pain, sometimes the pain's really hard at first, and then a few seconds later, it kind of calms down a little bit. That's because in response to pain and things like that, our body releases, and our brain releases a neurotransmitter called endorphins. Endorphins are the body's natural pain killers. And what drugs that are painkillers do, like morphine and Vicodin and things like that, they're actually endorphin agonists and cause more of that pain relief activity, to the point where people get really high and really addicted to it. So we'll get to that again in the next chapter. But if you've ever run a long way or exercise excessively, and at the end it almost feels like you're a little bit high, that's something we call runner's high, which can come from excessive exercise and, of course, in orgasm when having sex. That good feeling at the end is not just the release of your bodily fluids, but it's also your brain's release of endorphins that makes you feel really good. It may last a few seconds, it may last a little longer. We also have something called neuropeptide Y, which we'll talk about in chapter 6, and that actually tells your brain that your body needs to eat. And it also helps to produce shifts in your metabolism. And like I said, that will be in chapter 6 after chapter 3 in module 2. To make things even more complicated, and I'm not going to go into this because it's already been a lot here, we have gases like nitric oxide and carbon monoxide, which can actually help to regulate neurotransmitters, influencing the release of neurotransmitters from the presynaptic side. And if you ever wondered how Viagra worked, because I'm sure you all wondered, it actually enhances erections because it increases nitric oxide, therefore concentrating more blood flow to relax the blood vessels and allow the penis to engorge with blood and, do its thing. So that's your neurotransmitters. And if we were to look at it, right, so we have action potentials, we have chemical neurotransmitters, and in addition to that, we have networks of neurons that actually function together to form a circuit when we're seeing things and hearing things. In chapter 9, we're going to go over language. Why is it that we can read something and verbally respond? We can hear something and write down an answer, and it's sort of interchangeable. That's using circuitry from the visual system and the auditory system in order to communicate. So we have these networks. Memories are in a network. Language is in a network. There's a lot of things that are in networks to be able to process together. So many neurons working together in a circuit or network. Our neural impulses, we have more or less neural impulses. And when I talked about the rate law at the end of the first version of this lecture, I talked about how we know things are brighter or dimmer, how we know that sounds are louder and quieter. That's because of the number of action potentials we have. So trains of neural impulses can encode additional information in the intervals between the spikes and the length of burst. So seeing more action potentials makes a light brighter. Less action potentials can make a sound quieter. And some information is even encoded by segregating it into specialized pathways, which we call labeled lines. Don't worry, I'm not going to get into that. So overall, I'm just showing you exactly how complex the brain is. In the brain, information is integrated. The brain has to know what are excitatory, what are inhibitory, do we have enough to reach the threshold, and processed in complex neural networks. So one way of studying these networks is to try and simulate their activity with computer-based neural networks. Some of these things can work, like seeing when a person may get anxious or not, being able to detect cancer in a sample of a biopsy. But overall, the reason why we still use animal models is because as complex as the technology gets for AI and computer-based simulations, you can see where a computer will make one decision over another overall, but computers are not anytime soon, if ever, going to be able to simulate real thinking and decision-making the way a human brain can. Animal models on a smaller scale, more simplified scale, can do this. And this is why we still need to use animal models for biomedical research, especially when it deals with the brain. So we have gray matter and white matter. The white matter, as I pointed out, is these myelinated axons, which share the information and the action potentials through this. Here we see an image of the white matter tracks that connect the left hemisphere and right hemisphere of the brain, called the corpus callosum, and we'll go more over this in chapter 3. But for right now, we're going to skip to chapter 5 because we just talked about what the neurotransmitters are. So now we're going to talk about how they're affected, faster, slower, and what happens when we use drugs. These are medications, these are illicit drugs, these are anything from outside your body that causes the neurotransmitters to function differently than they normally would. Thank you guys for your time, and as always, I will talk to you soon.